I now call to order the Society's 2,490th meeting in what is now the 152nd year since its founding on March 13th, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends to tonight's PSW meeting and the lecture by Bartek Rajwa. The Society is grateful to PSW full-year science sponsors, PSW member Mike Helton and Helton Associates, LLC, for their support. And to the sponsor of tonight's lecture, PSW member Joel Schulman. Please join me in thanking them very much for their support. So I'm pleased to report that the 2-to-1 donor matching donation has been fully funded. And we're very grateful. We are, of course, very grateful to the donor who made this gift and to those who responded so enthusiastically. So thank you for joining me in applauding their contributions. I'm pleased to note that the following new members have been admitted to the Society. Jerry Hickey, a member of the faculty at Marymount University, interested in education and medicine, who learned a PSW from a PSW member. And tonight's speaker, Bartek Rajwa, whose interests will be clear to you in part from tonight's lecture. Well, he has a lot of interests. And he's very, very knowledgeable in all of them. If you're interested in membership, you can access a membership application on the PSW website by clicking on the blue Join button on the homepage at the upper right-hand corner. Or if you're here in the Powell Auditorium, you can access the application using the QR code on the cards at the tables at the back of the room. Please take some of the cards and give them to your friends and even people you don't know who might be interested in PSW. All members are entitled to a signed copy of Volume 1 of the Bulletin of the Philosophical Society of Washington, which is the original name of PSW. And if they choose, may purchase a ribbon of the Society, which they may wear. If you have not received your signed copy of the Bulletin, please let me know. Recording Secretary Scott Matthews will now present the minutes of the 2,489th meeting and the lecture by PSW members Lloyd Mitchell, A.J. Kothari, and himself.
On January 26, 2024, in the Powell Auditorium of the Cosmos Club in Washington, D.C., President Larry Milstein called the 2,489th meeting of the Society to order at 8:03 p.m. Eastern Time. He began by welcoming attendees, thanking sponsors for their support, and announcing new members. Scott Matthews then read the minutes of the previous meeting, including the lecture by Mike Neufeld, entitled Back to the Moon to Stay? The minutes were approved as read. President Milstein then announced that the format for the evening's lecture would include 3 short presentations by 3 PSW members, Lloyd Mitchell, Scott Matthews, and A.J. Kothari. The first speaker was Dr. Lloyd Mitchell of Retrotherapy, and his lecture was titled Gene Editing Using the Body's Own Editor, RNA Transplicing for Therapy. Dr. Mitchell began by introducing the central dogma of molecular biology. He described how a region of DNA is transcribed by an RNA polymerase to form a pre-messenger RNA, and how the used and unused regions, called exons and introns, are separated during the RNA splicing to form the messenger RNA that leaves the nucleus. This mRNA is read by ribosomes to create proteins. He indicated that the splicing can occur in many different ways so that approximately 20,000 genes can code for more than 100,000 proteins. He discussed traditional gene therapy, where both the complete gene and the gene regulation mechanism must be included in the delivery vector. He then presented the idea of gene editing, where only a portion of that gene needs to be rewritten and is delivered, and its expression remains under endogenous control. As a result, gene editing works with a much smaller payload and a smaller regulatory switch. Mitchell discussed the advantages of gene editing as compared to CRISPR-Cas9. He then discussed RNA transplicing, a technique which he invented, which allows genes to be altered at the RNA level where the splicing is happening. Mitchell explained that a trans-spliced gene can be delivered in such a way that it is permanent, usually delivered by a virus, or temporary, usually delivered by a plasmid. He then discussed recent laboratory results where the gene editing technique was shown to be effective in treating hemophilia, Huntington's disease, and cancers caused by the papillomavirus. The second speaker was Dr. Scott Matthews of the U.S. Naval Research Laboratory, and his lecture was titled Magnetic Mistakes in Their Correction. Dr. Matthews began by introducing the hysteresis branch crossing, a little-known magnetic phenomenon predicted on the basis of theory in 1935. He stated that by 1948, the magnetic research community had concluded that this phenomenon was impossible due to the erroneous assumption that it violated the laws of thermodynamics. Matthews said that the observation of the hysteresis branch crossing was not reported in the literature until his group published a paper in 2020, showing that it was a robust and reproducible phenomenon which violated no fundamental law. He then presented a brief lesson in magnetic materials with the goal of introducing three concepts in magnetism: magnetization, hysteresis, and anisotropy. If the audience could understand these three concepts, he said, he believed they could understand the nature of the hysteresis branch crossing and why people had jumped to the conclusion that it was impossible. After discussing these three concepts, he showed experimental data demonstrating the crossing and showing that it was in excellent agreement with a mathematical model called a distributed temperature-dependent Stoner-Wolffarth model. Matthews stated that both the experimental observation and the presented theory clearly showed that the prohibition of the hysteresis branch crossing was a mistake, and that mistake had now been corrected. The third speaker was Dr. A.J. Kothari of Astrux Corporation and was titled Thorium Reactors. Dr. Kothari began by stating that, quote, nuke is not a four-letter word, and that the coming climate catastrophe, because of the coming climate catastrophe, the modern world will need nuclear energy, and that he believed that thorium molten salt reactors constitute the best possible solution. He then compared nuclear power to fossil fuel technologies in terms of total greenhouse gas emissions, total land usage, and total number of deaths attributed to each technology. He compared thorium reactors to the pressurized water reactors, or PWRs, used for electric power generation, indicating that several of the hazards associated with PWRs are not present in thorium reactors. These advantages include low pressure in the reactor vessel, less high-level radioactive waste, and less risk of nuclear proliferation. Kothari then listed some of the details of thorium molten salt reactors. The thorium oxide fuel is dissolved into a fluoride-based salt. The salt contains lithium or beryllium. The salt is heated to about 450 to 650 degrees centigrade, and the salt can act as the primary reactor coolant. He stated that the U.S. has 600,000 tons of thorium, and that 1 ton of thorium can produce as much energy as 5 million barrels of oil. He described the thorium fuel cycle, in which thorium-232 absorbs a neutron and becomes thorium-233, which subsequently beta decays into protactinium-233, which again beta decays into uranium-233, a fissile isotope. Kothari indicated that thorium is 3 to 4 times more abundant than uranium. He ended his talk by mentioning that China is currently planning to build the world's largest container ship powered by a thorium reactor. The lecture was followed by a question and answer session. One member stated that, quote, RNA splicing seems too good to be true, and asked, what's the catch? Mitchell responded that pre-mRNA is not linear as depicted in his diagrams, but actually folds onto itself. As a result, it is difficult to figure out which parts of the pre-mRNA are accessible for base pairing, and this requires both bioinformatics and high-throughput screening. A member asked about Arriva, a French multinational group specializing in nuclear power, and their plans to implement thorium-based reactors. Cathari responded that he was not aware of any plans for thorium reactors associated with Arriva or the French National Electric Utility. He indicated that he was aware of thorium reactor plans only in Denmark and the Netherlands. Lloyd Mitchell, a member, asked A.J. Cathari about the Chinese cargo ship and when it would be functional. Cathari responded that the current schedule would have it operational in about 6 years. A question from the web asked Dr. Mitchell if CRISPR could be used to target an addiction gene. Mitchell responded, quote, "Undoubtedly. But CRISPR requires the Cas molecule, which is quite large, and so delivering enough to neurons in the brain might be quite difficult." A guest asked Dr. Matthews if he had a hypothesis as to why some materials exhibit the hysteresis branch crossing and others do not. Matthews responded that a well-defined magnetic anisotropy was required in order to experimentally observe the phenomenon. He stated that the anisotropy must have both a narrow magnitude distribution and a very narrow spatial distribution. After the question and answer period, President Milstein thanked the speakers and presented them with PSW rosettes, signed copies of the announcement of their talks, and signed copies of Volume 1 of the PSW Bulletin. He then announced speakers for upcoming lectures, made a number of housekeeping announcements, and invited guests to join the Society. He adjourned the 2,489th meeting of the Society at 10:01 p.m. Eastern Time. Temperature in Washington, D.C., 16.1 degrees Celsius. Weather, partly cloudy. Audience in the Powell Auditorium, 48. Viewers on the live stream, 37, for a total of 85 live viewers. Views of the video in the first 2 weeks, 369. Respectfully submitted, Scott Matthews, Recording Secretary.
Thank you, Scott. Are there any comments, corrections, additions, subtractions on the minutes? Hearing none, I will entertain a motion to accept the minutes as read. We have a motion to accept. Do we have a second? All members in favor? All members opposed? Hearing no opposition, the minutes are accepted unanimously as read and will be posted to the website in due course. If anyone online has any comments or suggestions or corrections, please forward them to corresponding sec@pswscience.org. They will be considered and the minutes will be modified as required, should it be necessary. We now turn to tonight's lecture by Bartek Rajwa on Spectroscopy of Deception, Unmasking Food Fraud with Biophysics. Bartek is research professor of computational life sciences and bioinformatics at the Purdue University Bindley Bioscience Center. His research focuses on biological data science, encompassing bioinformatics, biostatistics, and multi-omics data analysis, which he employs to synthesize information on a wide spectrum of measurement technologies, including proteomics, metabolomics, or metabolomics, genomics, biological imaging, lipidomics, and cytometry. He uses mathematics, computer science, and machine learning to understand complex phenotypic patterns, focusing particularly on automated phenotype classification in the presence of incomplete and noisy data. He co-developed with collaborators several groundbreaking technologies, including spectral flow cytometry, automated clinical flow cytometry analysis pipelines, algorithms for food-borne pathogen identification, and advanced spectral information processing methods for cytometry and imaging. He is editor-in-chief of Cytometry Part A, published by the International Society for Advancement of Cytometry. He earned a BS in molecular biology and biophysics and a PhD in biophysics. I'm going to say this wrong, and Jagiellonian University in Krakow, Poland. You can correct me later.
I will.
I count on it. All questions will be fielded in the Q&A session after the lecture. Bartek, the stage is yours.
Thank you very much for this generous introduction. So, today's topic is going to be food fraud. But before I move to food fraud, let me add a few things about myself, so you understand that I'm actually a human being and I have hobbies. A part of all of those scientific endeavors that I'm involved in, I love skiing. And when Larry invited me to give this talk, I was in Keystone, I believe, or Breckenridge in Colorado, and that is the picture which was taken on the day that he interrupted my leisure. This is a view from the new open area in Keystone, and you see Lake Dillon behind. We had fantastic weather, as you can tell. The picture was taken by my son, who is an avid photographer. So, as Larry mentioned, I received my education from Jagiellonian University in Krakow, Poland, before I moved to United States. And I would say I'm probably, or at least I hope, I'd like to be the second most important alumni of this university. The first one would be Nikolai Copernicus, who also studied at this at my alma mater. In fact, if you go to the university museum, they still keep the documents from the registrar's office, and you can see this huge book where they will open it for you and point to a line where it's written Nikolai Copernicus paid in full for the semester. Because obviously for universities that was always the most important thing, and still is, I must say. I moved to United States in 2002 after I was being recruited by my colleague and a faculty member of Purdue University. And I planned to stay for just a couple of years. And 22 years later, I'm still there. So when people ask me how long you've been at Purdue, I always answer too long. My work broadly encompasses all areas of data science, and what I'm going to present today is just a part of my interest, but it's probably the area that I really care on the emotional level most deeply, because we all eat food. So caring about food fraud and trying to prevent food fraud, I think that it's something that everybody can identify with. So before I move forward to the actual talk, I have to recognize my colleagues who assisted me, supported me, contributed to this research. And you see a number of names here, but I wanted to bring up actually Carmen, who was a student working with all of us. And I want to bring up Carmen because, thank you, because she's the link between this talk and one of the previous lectures that was delivered here, a fantastic lecture that I watched online on Artemis mission. So why she's the link? Because the methods, the biophysics methods that you will see that we are using and applying to the field of agriculture and food analysis, these are the methods which could be broadly applied to material science. And Carmen uses exactly the same technique that I'm going to introduce to you, working now for a company that designs the laser-based sampling system for Artemis mission. Yes, it's the same technology. People like me, they use it to sample cheese. Really smart people, they take this to Mars and do much more interesting stuff. And of course, I have to also mention, speaking of money, you see, I already forgot. I have to also mention that all of this research has been funded by really, really generous support from USDA and from the Center for Food Safety Engineering at Purdue University. We wouldn't be able to do any of this stuff if it was not for really visionary leadership of our USDA colleagues and administrators who thought that, well, it's really a good idea to use engineering systems and material science systems to analyze food in a way that maybe other people wouldn't consider applicable. You also see a picture of Purdue University and the map of United States there, and this is because I talk about my research on many European conferences, and recently I went to a conference in Prague, and for some reason, after they invited me and I was on the list of speakers, and I obviously identified myself as representing Purdue University. In all the program everywhere, I was shown as being from Canada. So I am trying to make sure that nobody thinks that Indiana is in Canada. And as you will see in few slides, it's very, very important that we don't mix Indiana with Canada. This actually matters. I care about it deeply. So, food fraud. So what's food fraud? Well, if you look online, if you check the definitions, you will see that they are quite complex. And there is USDA/FDA definition, which guides our regulatory agencies in United States. And there is considerably more complicated definition from European Union, as you would expect. Interestingly, the subject of food fraud is taken way more seriously in Europe than it is in United States. And I believe that there are multiple reasons for that, one of which would be not necessarily scientific or political or administrative, but rather, I would say it's an intersection of economy and culture. And that is, if you have economies which very deeply care about small manufacturers, tiny places which make the same products over hundreds of years according to one recipe, and they build the core of the local economy, and they contribute not only to the economy, but they contribute to the cultural identity of the particular region, then of course as a politician from this region, you would care if somebody commits food fraud and misrepresents product as coming from this region, right? So you all are aware that for instance, French would be very upset if you call any sparkling wine champagne, right? But it goes even further. It's true about cheeses. It's true about other local products. In US, the situation is slightly different, because sadly, and let's be honest, we care more about quantity than quality in terms of food. Unfortunately, this is changing now, and because it's changing quite rapidly, and it's obviously consumer-driven change, right? There is always push and a pull. So consumers are pushing towards better quality food, and regulatory agencies are pulling the producers, the food processing organizations to make sure that they deliver this quality. So this is changing now, but still, interestingly, if you look at the amount of, I would say, regulatory, well, it's hard to measure the amount of regulatory oversight, right? Amount of effort, maybe let me formulate it this way: the amount of effort which is driving the regulatory oversight, or even organizations which are tasked to police food fraud, you will see that Europe still takes it much more seriously. And there are a few steps ahead of us in all the areas that food fraud and food safety also could be researched, investigated, and applied. So, again, let's take the USDA definition. Food fraud is essentially an action which involves fraudulent, intentional substitution or addition of substance or a product for the purpose of increasing the apparent value of the food product or reducing the cost of production. As you noticed in the previous slide, the EU definition is slightly different. Well, first, it refers to a much more complex document, which I have not attached, but it also emphasizes the issue of intentionality, right? So we are not talking about just food quality in a sense that, yes, there is food which is better quality, worse quality, but the element of intentionality is very, very important if we talk about food fraud. And in United States specifically, and actually this is not only, I think it's not only US statistics, I suspect, I don't remember exact numbers, but I suspect that the order of the food products that are sort of targeted by fraudsters mostly is pretty much the same probably in EU too. So oils, right? Olive oil. We probably have heard about scandals when it's found out that what one thinks is olive oil from Italy, it's not really from Italy, and it's not even olive oil. Juices, spices, that's a big thing. All kinds of industrial colorant has been found to be added to spices. Use of artificial sweeteners, fake honey, all of those things, we are all aware of that. So this top group, this is around 70% of all the cases. And then there are other more specific issues which kind of erupt from time to time, and then they disappear from the headlines. For instance, a few years ago, it was a huge deal that the so-called Parmesan that you could buy or get in the restaurant was actually not Parmesan mostly. It was typically very old cheddar. And I'm not saying aged cheddar, as sometimes news agencies portrayed this, because it almost adds a level of legitimacy to the entire thing. Aged cheddar sounds almost good, right? No, it was just old cheddar, old rancid cheddar mixed with wood pulp. So that posed as Parmesan, and you could get this stuff everywhere. It's apparently #1 sold cheese in United States, right? Wood pulp and old cheddar. But a few people were arrested. The thing disappeared from the news. Then you see all of those news regarding spices. Many of those reports come from Europe, because, as I said, European agencies still care more about it. And I don't want to say that US regulators are negligent necessarily, but sometimes indeed the number of attempts to commit food fraud for given food product or given category of food products is indeed, the rate of those attempts is indeed higher in Europe than in the United States. So it's a paradoxical situation. We don't care about it that much, but fraudsters are not targeting US to the same extent. But as I said, the reason is the opportunity, right? If you take, let's say, cheese made in Wisconsin and you make cheese in some other state and slap a label Wisconsin cheese on it, well, you're not going to make millions on this, right? Maybe that's the stupidest scam ever. But if you take, let's say, really valuable commodities like Parmesan, Gruyere, Comté, then you can actually make a lot of money. So since opportunities are there, indeed, European markets are targeted more. And as I said, a part of those products which we see this type of news popping up from time to time, and then they disappear, and we are kind of worried about it, but not so much, because after all, nobody died from sprinkling some fake Parmesan on fake pasta in fake Olive Garden, right? I mean, it's not a big deal. It's a part of the experience, I would say. But I'm sorry, but from time to time the situation is much worse, and I want to bring up as a first vignette. I will have 3 of those. The famous horse meat scandal in 2013 in UK. So you may remember, or maybe not, well, everybody in Europe was following this, and obviously everybody in UK, but this was just, one of those strange scandals in a foreign country in 2013. But yeah, the case is quite illustrative and interesting. So how it started? Through routine analysis and testing, it has been found out beginning of 2013 that a large number of samples from 3 major manufacturers, food processing plants and manufacturers of food, samples which were supposedly containing beef, contained horse meat. And all of those manufacturers were in Ireland. And they were exporting this beef to UK. So they were not in Northern Ireland, they were in the Republic of Ireland, and they were exporting contaminated beef to UK. So you can imagine that the press was not very kind, and you can just right between the lines, you can guess what type of cover poor Irish got for this. But when the investigation started, and as I said, it started first with just those few manufacturers, but it has been found out that they supply massive number of large retailers in UK. I think that the first one that was found to be problematic was the samples which they got from Tesco, right? Which would be sort of like if you found a major contamination in Walmart. We are talking about a major retailer. So the samples came from Ireland. Investigations started. The Irish did their own investigation, and they said, well, all of this was actually not processed and packaged in Ireland. It came from Netherlands and Spain, right? So we are pointing to the Dutch and Spanish. They sent us the contaminated beef, or really horse meat, which was mislabeled as beef. But packaging and processing plants in the Netherlands did their own investigation, and they said, "Oh, no. We bought all of this in Poland." So that the Poles are really the culprits. By the way, the Tesco chain, when they ordered this meat, it has been the contract stipulated that it was supposed to be Irish beef. So even if there was no contamination, there was already breach of contract, because they were not supposed to subcontract it to the Netherlands, and Netherlands certainly was not supposed to subcontract it to Poland, right? But setting this aside, so we already know what happened, right? We have the meat that supposedly came from Poland, and maybe some from Spain, to the Netherlands, then from the Netherlands to Ireland, from Ireland to UK. So who is to blame? Now, this is my question to the audience. Who really contaminated this beef? Whom would you blame if you know that, well, it was an Irish company with the suppliers in the Netherlands, with suppliers in Poland? Who is to blame? Well, you would be wrong, and that's why I stipulated that we should be careful with geography, and I didn't want Indiana to be misrepresented. We have to blame Canada. So the investigation found out that the horse meat came from Canada, Belgium, and Romania. None of those countries were really involved in the scam. The products were mislabeled, so the horse meat was sent from those countries with a very, very different designation. It was supposed to be used to make dog food, right? And there was beef sent from Poland, which was supposed to be used for hamburgers. It was still obviously a fraud and a scam because they were supposed to be mislabeled as Irish hamburgers sold in UK. They were Polish hamburgers, but it was still beef. However, the horse meat from Canada, Belgium, and Romania kind of changed the labeling and designation upon arrival in the Netherlands. And what left Netherlands was already labeled as Irish beef for some reason, but it was not. Well, many people were arrested. It was a huge, huge scandal. And this actually triggered a total rethinking of the policies in all the EU countries. Remember, that was still at the time when United Kingdom was a part of the EU. And there was a very in-depth investigation led by Professor Chris Elliott from the Queen's University Belfast, who is probably the most prominent food safety scientist in UK and certainly in Europe. At that time, the lead food scientist in EU, and him and his team and people who work for the British government redesigned the whole system that was later on adopted by other countries. Since then, obviously, UK left the European Union, but to a large extent, the regulatory framework and the way how we think about food fraud or how they think about food fraud, and to a large extent also how US regulatory agencies think about it are informed by this experience. Because what was found out at the time is that, well, number one, there are not enough labs to test the samples. Nobody knows how to test the samples. There are no standards which they could follow or best laboratory practices to test the samples. There are multiple agencies which are responsible, and when one agency fails, the other agency doesn't pick up the burden, and at the end, nobody knows who is responsible for anything. And that interestingly coincided with the situation where some of the food processors, producers, manufacturers in UK were complaining they've been audited to death. So there was an auditing system in place which put a lot of burden on the producers and apparently failed utterly to deliver any results. It almost reminds me of the article I read in the hotel today when I arrived about the whole regulatory system for airplane safety. And somebody concluded upon the recent news that the installation of these failed doors, which exploded on the Alaska flight, right? That this actually happened in the Boeing factory, that they know for sure that the fault was there, and one of the people representing one of the regulatory agencies said, well, the way how we know that our regulatory system failed is that it didn't stop this occurrence, right? So that was exactly the same conclusion the regulatory agencies had in Europe, that, well, we had all of those audits, all of those agencies, but none of them stopped the biggest scandal from happening. And by the way, there is this huge overlap between the issues of just consumer, I would say, consumer trust and the economic factors and safety, right? On the last slide, I have to go back there. There's one thing I wanted to bring to your attention. Some meat had traces of, well, it's typically called in the trade, it's called just bute. Complicated name, people call it bute. So this is a horse tranquilizer. And you don't want this in your food, trust me. Well, unless you are into this type of things, but no judgment. Typically, people don't like to consume horse tranquilizers, and that was also found in the samples, which interestingly opens another possibility. Because many countries in the world, not many countries in the world use this particular drug in the veterinarian context. United States is one of them that does. So there has been a hypothesis, which obviously cannot be proven, that some of those Canadian horses were actually American horses, which were sent to Canada from where they went to the Netherlands, and then to Ireland, and then to UK. Yeah. So there is this overlap, right, between the food fraud, food quality, food safety, and what is often called food defense, which is essentially the whole system that we should organize around safety of the food supply chains. Therefore, if we care about food fraud, it's not that we just want to eat fancy food and we are snobs, right? It's really more than that, because typically when fraudsters engage in mislabeling, or if they engage in substitution, I would say that in most cases they don't care about other regulation as well, right? So you cannot expect that if somebody is running a production of a fake food product in his basement, this person is particularly concerned about, people wearing correct gloves or masks or protective systems, and he's probably not concerned about water filtering and air filtering and anything like that, right? Pardon? So, food quality and food fraud and food safety are intertwined, right? If we talk about one, we really talk about other. It also means that if we have a good system for detecting food fraud, we are very likely to identify possible threats in terms of, emergence of food-borne pathogens and stuff like this. And I want to underline this because very often there is a misunderstanding where when exposed to this type of research, some people say, well, okay, this is all great, but we have much more important things, rather than policing whether what you eat is really, let's say, ham from Parma rather than, it's produced by Smithfield, right? Well, you would think so, but no, because all of those things are interconnected. And there is obviously also the economic aspect, because if there is some group of organizations that cheat, it means not only that they are, cheating the consumer, but they are cheating their competitors, right? If you use, let's say, a product. If you use a supplier who supplies you ingredient that is, let's say, harvested in a particular, in a situation where particular farming practices are used, whether you care for organic food or fair pay or whatever it is, right? And another supplier falsifies the certificate. Then what happens? Well, somebody is making money, and the organization that was honest is obviously losing money. So it's not only important from the food safety perspective, it's important from the economic perspective, and it's important both for the other competitors in the field, in the space, in the market, and it's important for the consumers. Those estimates which you see are, I would say there is a lot of hand waving around those numbers because we don't have good policing systems. So we don't really know to which extent we are, whether we are capturing in our statistics 10% of the ongoing fraud or 2% or 80%. So there are those estimates from a number of organizations. For instance, the Grocery Manufacturers Association of the United States says, well, well, this is probably $10 billion globally, and 15 billion. Oh, it should be actually swapped around, right? It's they say it's between 10 to 15 billion in the US alone. The European agency says, well, maybe between 8 billion euros to 12 billion euros in EU, but then UN says, well, it's 15 billion worldwide. So as you see, those numbers don't really match because everybody has slightly different way of estimating those numbers. And the truth is, we don't really know. But on a personal level, I believe the judgment of Professor Chris Elliott, who in his calculation, and I don't know if he did them on the back of a napkin or an envelope or he has some better insight, but he said that the sort of inside conversations with European Union agencies led him to believe that the money being made on food fraud in Europe surpassed, a few years ago, all the money made on illegal drug trade in Europe. So we are talking about really big problem. And as I said, even though this horse meat scandal led to, I would say, re-engineering of the regulatory framework in many countries, we still face those huge administrative complexities which kind of contribute to the entire mess. Interestingly, in EU, this regulatory system, this whole labyrinth is probably less complicated. In US, you have at least 3 agencies regulating food, right? FDA, USDA, and National Oceanic and Atmospheric Administration. These are not the only ones. And they divide those tasks in a very peculiar way. And I'm always confused when I talk with my USDA colleagues, when I, for instance, write a grant and I say, oh, we are going to, deal with, let's say, contamination of oysters. Great, USDA supports that. Oh, we want to deal with the fact that there is a microplastic in the water and fish are consuming microplastic, and then microplastic gets to the food supply and then we eat microplastic. No, USDA doesn't deal with this. This is another agency because it's fish. Well, I thought that if you regulate oysters, you would regulate fish. Well, we regulate some of the fish, not all the fish, right? So it's extremely complicated. And I think that probably it will take a similar scandal to the one in UK with the horse meat for the US administration to realize that what they need is one agency that actually deals with food and maybe one kind of, the name would be food police, or one agency that has also the capability of actually arresting people and prosecuting people in this space. We are not there yet. So if we have a crime scene, right, we establish that this is a big problem. We have a crime scene. What do we do when we arrive at a crime scene? What's the first thing that police does? Well, they fingerprint everybody, right? So the answer would be, why don't we do fingerprinting of the food and food tracing, right? That would be a solution. And indeed, in 22, FDA decided that they will put a special burden on producers of some of the food categories, because if you provide additional documentation of the supply chain, if you keep this documentation with the modern technology, especially modern public key encryption technology and blockchain, you can actually, to a large extent, keep track of all of those things. And they created a list of those products which they would like to trace. But of course, and that was motivated mostly by the higher risk of causing foodborne illnesses, right? That was the motivation. It was not food fraud per se, like the fact that instead of one fish you will get another fish, or instead of one cheese you will get a different one, but because, for instance, you want to have a special care about how aquatic species which are used for food are being distributed and handled, because if you, for instance, get yourself infected with Vibrio vulnificus, well, you can die in 7 hours, right? So that's kind of serious. But the traceability in general, when me and my colleagues look at that, we thought, well, this is a great idea. If we could pair the traceability with tracking and fingerprinting, then that could actually lead to something. And very similar thought process happened a little bit earlier, actually, around the horse meat scandal. A very similar process happened in Europe too. And everybody started to talk about the food fingerprinting. And obviously, when we are talking about food fingerprinting, then that's the moment where the biotechnology, biophysics, analytical chemistry enters the picture, right? So these are some of the current technologies for food tracing, fingerprinting, and what I broadly call food authentication. Because once you fingerprint a particular food product, then presumably you can authenticate it, although this is not such an easy step from fingerprinting to authentication, because the part which is in the middle is machine learning. But we'll get to this in a moment. When you look at this table, and obviously I'm skewing a little bit this towards the methods that I use, but I think that it's mostly honest representation. Some of those techniques are extremely sensitive. They are very good, very reproducible. They perform fantastically, and in certain contexts, you cannot replace them. For instance, if you analyze wine or if you analyze whiskey, you want to use mass spectrometry, right? This is you cannot really do it any other way. However, those systems are extremely expensive. They take huge room, right? You basically need a whole analysis system, which is like a whole lab, very highly qualified personnel, lots of funding to build the database if you want to do fingerprinting. At this conference, which I mentioned, my favorite European conference for food safety, the one where I was identified as representing Canada, I go to this conference every other year, and when I went there first time, what absolutely impressed me, probably that was what drew me into food analysis from some other endeavors, mostly in trying to fingerprint different hematological diseases, which was much less fun, was one of the presentations where a food scientist decided that to help the whiskey industry, and he got fabulous grant from the Scottish Whisky Association. He's going to fingerprint every whiskey ever produced and stored. And he got a sample of every one of those whiskeys because they funded him. And what was great was that, he needed few microliters, right? At tops for his mass spec, but whiskeys are not sold in microliter bottles, so he got a bottle of everything that was ever sold or ever stored in Scotland. And when he showed that as one of his first slides, I sat there and I was thinking, why am I trying to build machine learning expert systems for cancer? This is what I want to do. Yes, so if you have plenty of funding and access to every possible sample of those valuable products, yes, you would use mass spectrometry, and when there is a suspicion of fraud, the sample will be sent to your lab, you will analyze it. True. However, that's not the scenario that we are typically dealing with. The typical scenario would be a USDA food inspector going to a food retailer or to a supermarket, to a food processing plant, and carrying a small device that would allow him or her to fingerprint the actual product, right? And you cannot do that with chromatography system and then mass spec system attached to it, right? Or nuclear magnetic resonance spectrometry system. These are not systems which you could use for this purpose. And then there are genomic methods. Well, they work great, they're fantastic. If, for instance, somebody tries to sell you steelhead trout instead of salmon, then yes, you can use DNA. But what about products that not necessarily include DNA, right? They could be still natural origin, but they could be highly processed. They may not need, what I meant is they may not include any actual DNA content because of the processing. So we cannot use the molecular genomic techniques. And that's why some scientists in this space, and I'm one of them, thought, okay, why don't we use optical methods? So we still want to use spectroscopy, but we want to use methods which could be miniaturized, right? And optical methods probably could be miniaturized. Many of them already are. And in fact, in this research, we use the technique which I called, and I mentioned this during the dinner to some of the members of the audience, that I often use technique which I call scientific arbitrage, where you take a method which is well developed in one field and you try to apply it to another field. And those fields may be completely unrelated. The beauty is that very often the solutions are already available. We just need to identify them and find them. So, what do we have here? Laser-induced breakdown spectroscopy is one of those methods which I would like to draw your attention to. And it's a very, very interesting area of application. And I kind of already let the cat out of the bag because I mentioned material science, and I mentioned that one of the students working on this ended up on Artemis project. So you can tell that, yes, there were other applications of this method before we started to analyze cheese with it. And the other one, very widely used in pharmaceutical industry, is specifically is the Raman spectroscopy, what I had in mind, but all the vibrational spectroscopies and fluorescence spectroscopies and near IR and so on and so forth, right? They could be easily miniaturized because of tremendous progress in optics and optical engineering over the last several years, mostly because of the telecommunication industry, right? So that's another case where we are taking solutions from one field and applying them to the other. Those systems, those methods, they have their own problems. They are not as sensitive. They may not provide you with absolutely super fancy complete picture of your product, as for instance with a mass spectrometry, but you can miniaturize them. They are inexpensive. The analysis is much simpler. So we decided to focus on those two, and we started specifically with laser-induced breakdown spectroscopy. So what's a laser-induced breakdown spectroscopy? I'm going to introduce this method because it's kind of obscure technique. It's not like everybody who was involved in scientific projects may know about it. I mean, everybody knows what Raman is. Everybody knows what fluorescence is. But this is slightly different. So laser-induced breakdown spectroscopy is a technique which relies on a phenomenon caused by interaction between hugely powerful laser and, well, the matter, right? The substance that which we are trying to measure. So if this laser is sufficiently powerful, it essentially will create a tiny plasma plume, or to speak plainly, it will burn a hole in the darn thing that you want to look at. But the hole is going to be tiny. It's going to be so tiny, so little bit itsy-bitsy that it doesn't really matter. And in fact, laser-induced breakdown spectroscopy is even used to analyze historical artifacts, like old paintings, right? Because they consider the amount of damage to be so microscopic, so minuscule, that musea allowed this technique to be used in this context. So you heat the surface with the hugely powerful laser, and then what happens, a plasma plume forms, and then it cools down and emits all this energy. And essentially, this is just a high-tech version of burning something in the flame and holding a prism and looking at the spectra. It's just amazingly high-tech version of it, right? Because you have a fancy CCD camera, a sophisticated monochromator or a system to create this actually separate different wavelengths, and you have a laser. But a part of this, it's not particularly complicated. It's essentially atomic spectroscopy, but in very basic optical setup. Now, Raman spectroscopy, probably everybody knows, but just to remind you, it's essentially a type of light scattering, right? When photons hit a molecule, typically we observe so-called Rayleigh scattering when the energy is not it's not transferred between the photons and the molecule. So what we observe after the light is scattered is the same wavelength carrying the same energy. In Raman spectroscopy, the situation is slightly different. Some of the energy can be transferred to the molecule, which absorbs this energy and essentially utilizes it, so to speak. It's obviously a simplification to increase its vibrational state, and then it returns it back. So we observe change or shift in the light that is scattered, and it could go different ways depending on many interesting additional conditions, but in the simplest case, we will see so-called regular Stokes-Raman scattering, and we will see the difference between the incident light and the light which we observe. And because this is dependent on the structure of those molecules, right? How they absorb this energy. It's a great system to look at organic matter. Right? It's a fantastic system for this type of purposes. This is why it's very widely, for instance, used in pharmaceutical industry to find fake drugs, right? Or drugs which have some adulteration. Okay, and the other thing is that those two methods are complementary, because, for instance, Raman is mostly used for organic molecules to detect them, to quantify them. LIBS, or laser-induced breakdown spectroscopy, it's really shining when you try to detect the compositional chemistry around the inorganic elements, right? So one very nicely complements the other. They are on similar price level in terms of if you try to buy the actual system. LIBS is probably a little bit more expensive because it's not as widely used, but again, this is not even close to what you would pay for, chromatography system plus mass spec system. So here is the example. Let me get immediately to the example. You see here, it's actually me holding an orange and scanning this orange with this device, which you see on the slide. And this device is a laser-induced breakdown spectroscopy system, which you can just go online and buy. And they were not developed for food science at all. In fact, the people who developed them were mildly shocked when we approached them and told them that we want to use it for this type of application. So here's the interesting story: why they developed this type of systems and why many people in US and abroad use them. So as I said, laser-induced breakdown spectroscopy is essentially a system for compositional analysis, right? So people who buy old scrap metal Then they need to separate, let's say, copper from, I don't know, bronze, or different types of steel, different alloys, different aluminum types, and this is what they use. They carry this thing which looks like a drill or a gun, right? And they go around the scrapyard, and they can do immediately on the spot elemental analysis. And the small thing has you cannot see it here, but behind there is an LCD screen. You would just shoot the thing, and it will tell you what is the composition of an alloy in the real time. It's absolutely fantastic. So the idea is, why don't we use that? And then we'll use different machine learning algorithm than the one they use. And instead of telling you whether this can should be recycled with these cans or with the other type of cans, it will tell you if your Gruyere came from Gruyere or it came from Wisconsin. So as you can imagine, when this idea was first brought up, most of the people thought that is absolutely insane. But that's exactly what we are doing here. In fact, we not only use the commercial system, but we also build our own system. And the reason for it is that, as I said, those commercial systems are optimized for particular type of detection. Not only in terms of the software, right? You can always change software, but in terms of the parameters of the actual optics, right? So the power of the laser, the pulse width, the laser spot size, all of that was perfected over the years to work for material analysis, but it was not optimal probably for food samples. And so we built our own system too, and we actually used both, and we compared both. This is another example here. You see, it's either my hand or my postdoc's hand, and we are measuring coffee beans, right? And here we are measuring. Actually, this is leaps. Here we are using, I think, because we build a system which unifies both. And again, that should be introduced later, but I'm already telling you the most exciting part. I think that in this case, we actually use Raman to look at pesticides at the same time. So when we started this project, we thought, okay, so we'll look at bunch of different samples, and we will see if this fingerprinting idea makes sense, if it works. And I specifically focused on samples that, if you go to those food science conferences, they at the time, they were not widely reported. So people didn't work on them, right? People at the time, when I started this project, worked on many other things. Olive oil was the most popular item because it's, as I said, number one commodity that it's faked. People worked on rice, on tea. A lot of scientists in China works with leaps on rice because of the contamination of rice with heavy metals specifically. And on some other commodities. So we picked commodities which were super important, but not many researchers pay attention to them. Plus, as I said, when I first came to those food science conferences, the reason why I started to come was that I work on a project of trying to identify food-borne pathogens. So my interest was in biomedical side of it. It was not in really food science part of it. It was more the issue of finding emerging pathogens. But what I noticed was that those, a part of olive oil, the other commodity that everybody was trying to figure out how we can tell the difference between real and fake was wine. Right? So I thought, well, I'm already too late to get into the wine space. Well, I have one paper written on wine, but that's a space which is really, really crowded. So if my fellow researchers work on wine, I will work on cheese, right? That is a great pairing, wine and cheese, and together we can contribute to humanity. So I picked cheeses, and that was our first commodity that we looked at, right? It was cheese, and then coffee beans, balsamic vinegar, olive extracts, and spices. And that was the number of samples that we put together in our first run of experiments. Actually, what I'm reporting here, it's not our first run. Our first run was mostly focused on cheeses. It was a different instrument, different setup. But when we did a number of follow-up studies, we already looked at cheeses before. So we thought, yes, we will add those other things. And we also wanted to test if we can look at different, I would say, physical representation of a sample. So we wanted something that is solid, we wanted liquids, and we wanted powders. And this is our list of samples, and I'm not going to bore you with details. And however, I wanted to underline a few things. And again, I'm going to focus on cheeses. I could talk about every of those commodities, but what I wanted to underline is if you are a cheese person, so to speak, if you like fancy European cheeses, you will immediately recognize that they all belong to a very, I would say, narrowly defined category. It's not like I'm trying to tell a difference between brie and cheddar, right? I don't need a fancy equipment to do that. I can just look at the product. These are cheeses which are extremely similar, and some of them differ by, let's say, 20 kilometers on the Alpine road from one village to another, right? So we are talking about products which are very, very close. The very fact that they have differences in terms of perceptual differences, they taste differently, right? I can recognize them by taste, but obviously that's not a method that it's necessarily deployable everywhere, right? Just like with wine, the very fact that we can tell those super tiny differences in the origin is remarkable by itself. It mostly stems from the fact, and this is a digression, that's not the topic, but that's another interesting topic which I could talk about for hours. Different microbiome flora is found in different regions in the places where those cheeses are manufactured. And this microbiome actually not only differs between one village in Alps and another village in Alps, it also differs from month to month to month. So I can tell the difference whether the cheese is correctly formulated by using milk from cows that graze in the spring, from the cheese that is made from milk from cows that graze during the other months that, for instance, Association of Gruyere producers would not approve of, right? So the terroir, the microbiome, all of that contributes to the way how the cheese I would say not grows but matures, right? And then you have differences in the chemical composition. And on the top of that, you have differences in matrix in which this chemical composition is sort of embedded. And for spectroscopies, this is a major problem, so-called matrix effects. The same material represented in a different matrix, like whether it's powdery or whether it's more wet or whether it's more airy or has some other physical properties of the matrix which are different. For spectroscopies, this is a major problem, but for us it's not. Because spectroscopists try to normalize for it, try to eliminate the effect of the matrix. For us, it's not a problem because the matrix is itself representative of the original food product. So what we are trying to do, and again, I could talk about other products the same way, but the bottom line is we tried to put the bar extremely high. Right? Extremely high. We are not talking about, can I tell a difference between fake Gruyere from Wisconsin and the original Gruyere from Switzerland? No. We are talking, can I tell the difference between Gruyere from this village and that village? And before I go there, I will introduce you to another vignette and another justification why we also looked at spices. Because spices are very commonly faked too. And one of the biggest scandals, again, this unraveled in Europe, but if you Google for reports from United States, you will find that some of those problems were found in US too, is addition of colorants, industrial colorants to spices. So Sudan red was first found in Chile, in Europe. And this is not the stuff that you want to eat, unless, as I said, you like the horse tranquilizer, then chili with Sudan red is probably a good thing too. But for everybody else, no, it's highly carcinogenic. It's a thing that you don't want in your food. But there are more and more. There were more and more cases over the years where manufacturers added Sudan red or other nasty things to spices. And again, it's a question of opportunity. It's a question of prices of spices on the market. For instance, the joke in food safety industry is that, well, there is no really saffron anywhere, right? You cannot buy saffron. It used to be that you could buy honestly fraudulent turmeric. But there is no turmeric anymore because all the turmeric is faked too. Right? So it's some other thing which has nothing to do with turmeric. And in fact, we were telling this to each other in the food safety group as a joke, right? That sooner or later you wouldn't be even able to buy fake saffron because somebody will find out a way to fake turmeric. And over a year ago, there was a case in India where many people landed in a hospital because it occurred that the fake saffron which they bought was not faked by adding turmeric, it was faked by adding some industrial dye. So they ended up in a hospital. So those cases are happening. Anyway, coming back from the vignette, that is the justification why we also included spices here. So we have cheeses, we have coffee for obvious reasons. And as you see, not only we have different physical form, but kind of different justification. Because in the case of cheeses, it was, well, can we authenticate, can we really fingerprint products which are close? In terms of coffee, can we look at different varieties, right? Or products from different producers. In case of spices, it was, again, a slightly different justification, which I just introduced. Vanilla extract, it's obvious, right? Whether it's true vanilla or vanillin. Okay, and finally, we have our spectra. So we got this type of spectra when we first looked at it, and this is obviously not from the first experiment that we ran, but if you look at the spectra, they look tremendously I would say boring, irremarkable, just, okay, they are, and they all look the same, roughly speaking, with few peaks more here or there, but at the end, it's not like you can immediately look at those spectra and say, oh, okay, so I can see the difference. Why they all look the same? Because this is chemical composition of organic material, and we are talking about food. So it's all going to be almost the same. But this almost, not exactly the same, is enough to tell the difference. Because what happens is, you very quickly, when you do the analysis, you notice that those major peaks, although they are in the same place, because obviously all the food will have carbon and calcium and sodium and potassium, the proportions between the peaks is where the information is. And obviously one way to do this is go after those proportions and try to analyze them. First identify the interesting peaks and then analyze those proportions. But that's not what we did. This is averaged from two different systems: the handheld LIPS system and the benchtop system, and the same thing, right? You just see a bunch of different peaks, which kind of they are the same in every product. What we instead did is we used fairly simple but very powerful machine learning method, where we decided to identify the specific combination of peaks that are most predictable, right? That actually drive the prediction. So we built a model which is based on an old idea of regularizing multinomial regression. This sounds complicated, but the idea is actually very, very simple. You have a regular regression, and as we know, all the machine learning is just glorified regression, one way or another. So you take some kind of a regression formulation, and you build a model, but you build a model intelligently. You penalize the model for incorporating the features which are not predictive, and you kind of give a prize. You praise the model for incorporating features which are important and predictive. And you do it, obviously, through a particular mathematical formulation. So at the end, you may start with the whole spectrum, which may have thousands and thousands of numbers. And when you go through this process of regularization, you may end up with just few numbers that really well predict the category to which given product belongs and from which those features were calculated. And this is the result which we got. So as you can tell, and I hope that you can tell that, yes, this is big enough, is you can tell here we have those importance scores, which are essentially scores assigned to different features. And we take only the highest importance scores for our model. We identify them as being linked with particular wavelength. And if we can link them with particular wavelength, we can link them also with particular chemical compounds. And we do it with, actually, I lied. It was not one method. We used two different methods, but it doesn't matter because we try to do it in such a way that we can reconcile those two and pick the best predicting features. And then at the end, we can identify what is really driving the prediction for any of those categories. And we know that, for instance, for cheese is going to be iron and sodium, right? Or we know that for coffee it's going to be calcium and iron. And these are not the only elements, but these are the most predictive elements and their ratios, actually. One interesting thing, I don't know if you noticed, this is probably something that you wouldn't expect on this list, vanadium. Is it okay to have vanadium in your spices? What do you think? Oh, absolutely yes. The best source of vanadium, which is a natural microelement in the small quantities, is black pepper. That's where vanadium comes from. It's black pepper. So if you have black pepper that doesn't have vanadium, it's probably not a black pepper. It's something else. And that's how you can tell that you really have black pepper. But of course, you don't want vanadium anywhere else. Only in black pepper, because in black pepper it's naturally occurring microelement. If you have vanadium from somewhere else, then maybe those spices were growing next to some nuclear reactor. I don't know. Other members of the society know better about those nuclear things, so they may know. But yes, so sometimes those elements are unexpected. And if we plot one element versus the other, we can see an interesting thing, that for certain categories, taking just two top features, just two elements, is absolutely not sufficient to perform classification, right? We need to have this much more sophisticated mathematical model, for instance, for cheeses. And the reason why is because, as I said, those cheeses were so close to each other. So we cannot take just two elements and say, oh, I can tell one cheese from another. But for vanilla extracts, we just need two elements, and we can tell not only the producer of your vanilla extract, but whether it's really vanilla extract or artificial vanillin, right? So there are different results for different food groups. That is an important observation. And we also saw that for some categories, even with our best system and best mathematical models, those accuracies of fingerprinting are going to be lower than we would like to be. And again, I'm bringing cheese and coffee, and the reason is that they're all so close, right? So we were able to achieve accuracy of prediction, 84% with one system, 84% with the other system. And we wanted to increase this. And that's why originally we decided to incorporate Raman in this whole picture. And then we found out that we can do lots of other things with Raman. But that was our first reason for incorporating Raman. We saw that those more complex food matrices and more complex foods, especially those that come geographically from very similar regions or of the similar type, they are hard to fingerprint. And obviously, I'm making this shortcut, right? They are not hard to fingerprint in a sense that it's hard to collect the information. We can fingerprint them, but telling this fingerprint apart from the other could be hard, right? Okay, so we decided, why don't we combine this with Raman? And the first time we looked at this Raman spectra, we had the same observation. Oh, it all looks the same. It's not going to be particularly informative. Different type of cheeses. For instance, here you have different type of cheeses. Probably nobody would be able to tell what are the differences in this spectra. But again, just like before, we found out that, these are cheeses and coffee beans, again, that with this additional data filtering, additional modeling, we can actually do pretty good job. And data fusion, combining particularly well-selected LIBS features, spectral features, and particularly well-selected Raman features allows us to boost the fingerprinting of cheese recognition to nearly 94%. And we use the system described before, and we use the features extracted by the system with another very popular old-school machine learning algorithm called support vector machine. So we were able to tell with the probability around 94% whether your cheese comes from this side of Alps or the other side of Alps. And I think this is fantastic result, given that that was really an exercise in pushing the method to the limit, because obviously if deployed, it wouldn't be used this way. It would be used to those cases which are described. Is it Parmesan or old cheddar with wood pulp, right? And for that, I'm not even reporting the results, because this problem for those systems is so trivial that I always get 100%. So that's not a challenge. Okay, and this is the actual confusion matrix where, where you could see how every cheese was classified into every category and where were the misses, and the same with coffee. So when we had a Raman spectrometer in our hands, we realized one thing, that the cultural practices in different countries could contribute to the fingerprint. Right? Because different countries, for instance, have different soils, right? This is how mass spec people recognize that most of the French wine sold in China is not French wine, right? In fact, in China, every year there is more French wine sold, or labeled as French wine sold, than French wine produced in France every year, which kind of tells you that there is a problem there, right? And they can tell because the type of soil and the micronutrients in soil, microelements in soil, leave the chemical fingerprint. We cannot do it this way, not with LIBS, because the sensitivity is not there. But if we, for instance, look at global pesticide use, you see that different countries use different amounts of pesticides per area, per arable area, right? For instance, on this list, this is from over a decade ago, but on this list you can see huge difference between Belgium on one side and Baltic and Scandinavian countries on the other side, right? There's a very, very huge difference. And obviously the trace amount of those pesticides are left on the products. So not that we would use it necessarily, because even the trace amounts which are left are supposedly safe for consumption, so this is not a problem of food safety per se, but it's a fingerprint that is left, right? So we would know that if we have LIPS signal and then we have Raman signal which identifies presence of a particular pesticide, we could say, oh, the product is from a different location. So that's what we decided to do. The other thing is heavy metal contamination. And here the situation is a little bit more complicated because in general, yes, you can use it as a fingerprint, but you don't want to have any heavy metals in your food. So this is a fingerprint which would be necessarily associated with the bad stuff. And that could be detected by LIPS, actually not by Raman. And then we found another thing. Well, it's not us who found that. We found that we can use another thing, which is enhancing the LIPS signal and Raman signal by incorporating nanoparticles and creating a situation where there is a resonance between nanoparticles and the actual substance that we want to recognize. And this is called essentially nanoparticle enhanced LIPS, or and you can do the same thing with Raman as well. The method is called SIRS. So the way how we did that is, and that's where those pictures came from, we measured surface of fruit and we contaminated the fruit artificially with pesticides, but then what you do, you spray it with the nanoparticles before you make measurement. And we found out, and again, that's the same process, we found out that we can recognize both metals, not necessarily heavy metals. We didn't want to spray cadmium in the lab, but some other metals, and we can recognize presence of particular fungicides, pesticides, and other stuff. And the signal, indeed, and again, I don't want necessarily to ask you to analyze with your eyes those spectra, but you would probably recognize immediately that the level of the signal here is dramatically lower than the level of the signal here after enhancement with nanoparticles. And then we start to see those peaks from different metals. And that allowed us to identify the presence of those metals or particular pesticides or fungicides on the surface of the fruit. And we did that, as I said, it's a combination, because for metals you use LIPS, for pesticides, fungicides, insecticides you use Raman, and that's why we wanted to build a system which have both things built in, right? So there are no two, you're not like walking with two of those, but you have one, and it takes care of both. After that, we decided that we'll be even more ambitious and we'll see if we can quantify that. So not only the presence, but what is the amount, right? So obviously you recognize these are just calibration curves. So I'm just showing that we indeed made the effort and to look at the different peaks and look how they change over different concentrations. And we found out that to some extent this is possible with those methods. It's not perfect. When you look at the combination of the results and you look at what EPA, for instance, mandates, you could see that we are close to be in the correct ballpark. But again, this is not the method that was intended to substitute full lab analysis. The idea is that you're a food inspector, you have this system, you quickly inspect something. If something is probably wrong, then you send the sample for full analysis, right? It's not like the lab in a box, which can do everything. It will do authentication, it will do fingerprinting, and it will tell you that maybe you should look into food contamination with pesticides or insecticides or heavy metals. After we did all that, we thought, okay, so everything seems to work fine. Let's push the boundaries even further. Contamination with microplastic. Right? There are all kinds of microplastic now in the environment. Plankton eats microplastic, fish eats plankton, we eat fish, or other fish eats fish. At the end, microplastic is everywhere. It's nasty. There are all kinds of issues with it, and probably it would be good to have another speaker who just talks for 1 hour about danger of microplastic, because it's a fascinating topic. I will stop just by saying that this is important. So we use the same system with microplastic, and here the results are more problematic because obviously if we have contaminated surface with microplastic, for instance from faulty packaging or something like this, the contamination is not going to be uniform. So now, depending on where we hit this with the laser, we may have a situation where we hardly see anything or we almost see nothing, to the situation where we recognize the characteristic peaks which are associated with a particular type of plastic. And again, we tried it for different types of microplastic, and it's the same result. So unfortunately, this is not the best method to do that. It's a free information which you will get if you do the measurement. So you can do fingerprinting, and you may find plastic as well. But it's not the best method for finding the microplastic inside or on the surface even of a food product, because you would have to scan the entire thing, right? You may have microplastic in a particular region of your product. So yes, we can find it. When we stumble upon it, we can see it, right? Like for instance here, this is one of those is the actual calibration sample. I believe this one, or maybe this one. But you could see that when we stumble upon the bad stuff, we can see it. But if we are off just by a few millimeters, we cannot see anything. Okay, so the last thing is the future. What are we going to do in the future with all of this? And to justify our choice of the future research, I have to bring the very last vignette of the evening, which is Chinese milk scandal in 2008. And again, you may remember this from the press. The powdered milk was contaminated with melamine. And melamine is this high nitrogen content product that it's additive to plastics, laminates, adhesive, and so on and so forth. But because it's rich in nitrogen, it fools some of the tests for protein content, right? Because if you test for the content of protein in milk, let's say, to see if the milk is diluted or not, and there is a melanin in it, if your test naively assumes that all the nitrogen comes from the protein, which is normally occurring proteins in milk, it will be fooled by melamine. And now, kind of everybody knows about it, so we are not expecting that this type of scandal will erupt again, but that's our motivation. This 2008 situation, which led to a number of deaths, is our motivation because there is other product which is not like powdered milk, but it's close. These are all the protein mixtures, additives, protein packages for athletes. And we haven't seen any reports of them being contaminated by melamine. But what I found out is more and more concerns about contamination with heavy metals. And we thought, okay, we are not going to be studying powdered milk because there is thousands and thousands of scientists in the food science area that already has that job to do, right? Everybody knows that because of the melamine situation, we have to monitor powdered milk, but nobody seems to be monitoring those protein powders. If melamine suddenly pops up in them, nobody would know. And we already know that people found arsenic, lead, and all other kind of stuff. And laser-induced breakdown spectroscopy would be absolutely perfect method to detect this, right? Because these are heavy metals. So these are our future plans. Okay, so let me conclude. I introduced to you two methods of spectroscopy, which were really taken from other fields of science, from material analysis, from analytical chemistry, from biophysics. They were not meant to be used with food samples, but we found out that they are actually a perfect match for working with food samples. We build a system which combines both, and we can recognize all kinds of interesting food contaminations, and we are going to continue this work. This is my postdoc, Sang-Ho Shin, who who worked on this project with me. He concluded his postdoc time in United States and came back to Korea to look for opportunities in industry. But I have to recognize him. He's a great spectroscopist and a good colleague, and he contributed immensely to all this work. So in the future, we'll be monitoring the food which you eat, or the food that your children and grandchildren eat when they, for instance, go to the gym and use those protein powders. I hope that we won't be able to find anything nasty there, but regardless, it's just a fascinating subject from my point of view, not only from the purely analytical perspective, but also because it integrates multiple disciplines. My personal favorite in this whole mix is one which I haven't spent too much time talking about, which is machine learning, that is involved in this analysis. But ultimately, it's a very interdisciplinary study that brings together people from food science, agriculture, biochemistry, biophysics, analytical chemistry, and other disciplines. And that's why I find it fascinating from the purely intellectual point of view, but it also happens to be important for the entire society. Thank you very much.
Well, we have time for a few questions. Most people know the procedure here. We have two mic runners, Brad and Pierre. Oh, no, we have two mic runners, Brett and Anne. So when I call on you by the color of your microphone, please stand up and tell us your name and tell us if you're a member. You do have to keep your hand up long enough for the mic runners to get to you. Ask a question, don't make a speech. Red microphone.
Maria Zemankova, member. Fascinating talk, because as you said, we all eat food. And I might be bad, but I really don't buy organic food because I don't trust it, that it's really organic. So can we really believe that one of these days the food is going to be truly labeled as organic versus not? It's, I have a million of questions, but this is probably the one that...
Yeah. That's, I must say, I could talk probably for hours just on this subject because it's one of my pet peeves. A lot of those labelings or organic farming practices, sustainable farming practices, all of those are not really regulated in a sense how we think about regulation, right? So it's really extremely difficult to know what is exactly there. And we also know that, at least as far as food safety is concerned, and I don't want to badmouth organic food manufacturers, but as far as food safety is concerned in terms of contamination with food-borne pathogens, unfortunately, organic manufacturing practices have a really bad record, right? Many people died, actually, from really, really nasty foodborne pathogens. And there are multiple reasons why. It's mostly unregulated industry. I hope we will get better. I also hope that methods which can fingerprint food can lead us to development of some kind of protocols and practices which would tell you if, for instance, particular type of pesticides or insecticides or fungicides were used or not. But one bigger thing is that we are thinking about organic food often in the context of, well, what is the biggest sort of worst thing that can happen when we eat commercially available food? Well, there are all of those chemical additives, right? But let's take this under the consideration. The biggest issue in terms of food contamination are not those additives, are the natural nasty products of bacteria and fungi, right? All the mycotoxins, they are killing people all around the world. And that's the problem which we have to solve first. On the other hand, I understand the concern that we put more and more compounds into our diet, which we don't really know how they interact with our immune system, with our endocrine system, and so on and so forth. And this is very serious concern. There's no question about it. Yet, if we don't have a good methodology to assess what type of farming practices were involved in producing particular type of food, then to a large extent we are engaging in, sort of cargo cult. And we think that if we buy a certain product which somebody labeled in a particular way because they for instance align otherwise with our worldview or political orientation or anything, then the product will be necessarily better. No, it has to be verified by scientific methods, and I hope that methods like this one and some other methods which are being developed will be capable of doing this. We are not yet at this point, unfortunately.
Let's go to the blue microphone and then the red microphone.
I'm Federica Dorima, a member. Thank you very much. Very fascinating. I never thought that there was so much kind of spectroscopy and Raman scattering and everything going into food kind of monitoring. My question actually is in the other spectrum of the organic, the engineered foods. As they kind of becoming more ubiquitous or will becoming more ubiquitous, what are the your methods, how they apply that, or how would they be used, both for people that want to avoid engineered meats, for example, versus those that consider it more eco-friendly and they want more? So where is the kind of the space for your work there?
Right, so we can understand the term engineered foods two ways, right? So these are bioengineered foods, which would be genetically modified foods, or engineered foods, which would be what people derogatory term fake beef, for instance, which is fake, not in a sense that it's a horse meat, but it's fake meaning that it's grown in the lab, right? So as far as the first type is concerned, then yes, this is easy because any genetic testing can recognize those products, right? If there is a modification in the DNA, so we can tell that that was a genetically modified product. Whether it's really a big issue in terms of all kinds of negative things that people attach to those genetically modified foods, whether this is really justified, that's a completely different topic. And in many areas, it's actually very unjustified. But from the perspective of analysis and fingerprinting, it's, as I said, easy because the very fact that they are genetically modified, it means that we can recognize them readily with methods of molecular biology. Now, as far as all the other foods which are engineered in a sense that there is a very high level of food processing involved. So there could be additives there, that could be colorants, that could be different packaging processes which are quite unusual and which we may not want to endorse. For instance, like use of a particular plastics or use of other things. Then these are the areas where biophysical methods obviously would shine, right? Because as far as you have another additive or you change the texture or you essentially reconstitute in the lab something which should look like cheese or look like meat, it's practically impossible to get those proportions exactly right. Proportions in a sense of proportions of different chemical compounds exactly right. And how do we know that it would be almost impossible? Well, as I said, you may have your cows grazing on grass just few miles from the village of Gruyere, right, and grow your cheese there, and this is enough to tell a difference. So it would be hard to believe that somebody can just from the first principle rebuild something that looks and smells and tastes like the actual food product and would be not detectable. So to sum it up, If the engineering involves methods of molecular engineering, then it would be the sphere of molecular biology, and we can do it quite easily. If this is engineering in the sense of building sort of fake products which have all kinds of ingredients which we may not necessarily want to consume, then actually combination of laser-induced breakdown spectroscopy and Raman would do a terrific job. And if we cannot do that, if we still cannot recognize certain things, that there's absolutely no way any engineered food product can go through the test which involves mass spectrometry and pretend that it's not fake. So the only issue is the cost, right? Because those are portable devices. You can take them on the road and you can test things with mass spec. We would need to send those samples to the lab, but this is all doable.
Red microphone, and then we'll go to the questions from the live stream.
Very good talk. I'm Kristen Ferry, and I am a member, and I'm a retired cattle and sheep farmer. So I could ask a zillion questions, but I think there are two things that I want to home in on here. One is that we have had a huge fight in the meat producer community over country of origin labeling. And it's gone back and forth. And unfortunately, beef got exempted from a requirement after it was instituted. And then there's this product of USA label that's put on, and most Americans will pay extra for that label, not realizing that, especially in the case of beef, the only thing it had to do to get that is to pass through a U.S. packaging plant. And most lean ground beef has an extremely high percentage of imported beef in it. The USDA is changing this, and it's very big flap going on, but it's supposed to become a label that says born, raised, and slaughtered and processed in the U.S. Is there some work going on to help us validate this label and prevent cheating, or give us an extra security about this label when it goes into when the new comes into effect? And another area that you haven't mentioned, but I'd like you to comment on future, is natural fiber and identifying, because we have a lot of counterfeit going on in cotton and wool.
Right.
And so if you could...
Yes, so I'm not aware of the regulatory effort around that, because I'm not following very, very closely the policy. However, I can offer the following insight. So working in this field, I notice extremely creative methodologies that people try to apply to solve the problem of the place of origin. Sometimes you cannot simply test the food product itself, but you can test other things which are, I would say, related or attached or present on the food product. I will give you one example, which is, in my opinion, quite interesting. So colleagues of mine from Purdue thought about the idea of testing origin of certain vegetables. And they look at number of things which you could test, right? You could test DNA, you could test all kinds of things. You could do one of the things which we do. But they found out that absolutely ingenious and spectacular and fantastic way of testing whether, for instance, your lettuce came from California or from Mexico is to take a sample of these remnants of soil, which would be at the bottom, and test for the genotype of the soil bacteria. So you can do absolutely ingenious experiments with things that even people who try to fake it, not that it's actually a big deal or a big economic problem of somebody trying to pretend that the Boston lettuce is from one place or the other, but just as a thought experiment, right? Probably fraudsters would never think that, well, just a little bit of soil which is on the roots, right, would be enough to identify where the product came from. So I think that this type of methodologies should be incorporated into arsenal of techniques that the regulators have. I cannot imagine that they would do it routinely with every product that shows up in the port of entry to verify the food origin, but a combination of occasional audits using biophysical techniques, biochemistry techniques, molecular biology, connected with another fascinating way of preventing this, which is traceability enhanced by public key cryptography and blockchain, right? This could actually lead to dramatic improvement in the ability to trace where our products actually came from. Right? This is indeed possible. And you can combine this with the blockchain and public key encryptions two ways, not only by representing what was sent to which producer and so on and so forth. You could even encode and encrypt the genetic fingerprints or biochemical fingerprints. So you would know that, okay, there is the fingerprint of this product when it was loaded on the ship in Genova, let's say, and this is the fingerprint of this olive oil in New York, right? And we have encrypted version of it in blockchain, so we can identify, somehow magically between Genova and New York, the olive oil from Italy changed into olive oil from Morocco, right? So with this type of methods, we could be one step ahead of the fraudsters, but it's a combined effort, right? So it's not only methods on the chemistry side or biophysics side, it has to be combined with the regulatory framework and with informatics.
We're going to take a couple of questions from the chat on the live stream, and then the last question will be Al with the red microphone. No.
Okay. I have 3 questions. Is that okay? So I'm going to start with a question from Mike Helton, who is a member. He wants to know, is it a consideration to correlate any of these measurements with human taste or smell?
That is a very interesting question. So there is a lot of work in the area related to analysis of wine. And I believe that also some people did the work with olive oil, which try to correlate certain perceptual descriptors with mostly with mass spec. Actually, I must say I'm not aware of anybody trying to build the correlates between laser-induced breakdown spectroscopy or Raman and the perceptual readouts, so to speak. But a lot of work has been done between mass spec, and there is a lot of papers published on the subject. So I would recommend that the person asking the question simply Google's in Google Scholar for work on mass spectrometry and wine and olive oil and quality and taste, because not only they try to link it with the particular descriptors, perceptual descriptors of taste, but also link those with the notion, perceptual notion, obviously, of quality, right? What people consider to be the good wine. Because not only you want to know that, oh, this wine is going to taste in the way that people characterize it as this, this, and this. You want to know after the mass spec analysis that this wine is going to be the good wine, right? So they try to build this type of things, and there is a lot of effort in this field. It's actually quite fascinating subject, and these are very advanced studies. A lot of effort has been put into this. There is a barrier of, I would say, the biggest barrier is the cultural barrier, right? We want to have a conversation with the sommelier. We don't necessarily want to be given a printout from the mass bank and say, well, this is what it is, sir. What would you like, right?
Next question from the chat.
Sure. Second question is from Jim Talons. He did not indicate whether or not he is a member, but he wants to know, is ramen or libs used at all by local authorities to check on restaurant substitutions such as shark instead of scallops in Asian restaurants?
Oh, okay. So none of those methods are routinely used. This is all development type of work. USDA funded our work, but this is not on the level. It's not advanced to the point that USDA authorities or FDA authorities would be using it to actually inspect the establishment. So the way how they do it now, and that's why they funded it, because this is something that hopefully at some point would be changed, is when the inspection happens, they take samples and they send it to the lab, right? And then a week later, you know what's going on. Now, coming back to the substitution, especially with fish. So the substitution with and food fraud in fish industry, this is a huge problem. And there is both substitution because you basically catch a bunch of stuff and you say, well, we mostly were looking for this type of fish, so everything we caught is, let's label it the same way, right? So that's one type of a problem. The second type of a problem is actual fraud. Right? When you, let's say, as I mentioned before, if you sell steelhead trout as salmon, it's fraud. The reason why people would do that is not that, by the way, there's nothing wrong with steelhead salmon, with steelhead trout. In fact, I prefer the taste. But if somebody charges me twice as much pretending that it's a salmon, then, it doesn't taste as well anymore, right? So, but those things, the best method to combat the food fraud in this space would be through DNA testing. And that's how this is typically done. And obviously, this opens opportunities to develop portable systems which would do like real-time LAMP or PCR or one of those fast amplification things, right? But I would rather do it this way than through this type of methodology, because typically we kind of know what people are substituting, right? It's, in fact, there is, if you go to, I believe, FDA site, they have actual table where they tell you which type of fish is most likely substituted by which other type of fish, right? So the testing would be, is it A or B? Right, and fast DNA test or something which is almost the type like the type of assays. Oh, I forgot the term for this type of assays, like the assay which we used for lateral flow assays, right? Like the one which we used for COVID, right? You could basically put a drop of some solution and it would tell you is it A or B. This is possible right now. I don't know if food inspectors are doing this. As far as I know, not. I think they are still sending everything to centralized labs.
Last question from the chat.
Okay, YouTube username UStreetUniverse has a question regarding FDA labeling requirements of food. He says, I am seeing more products which do not identify the source, but refer consumers to a QR code or website. I was wondering if you had anything to say on that topic.
Yeah, well, again, this is a policy question. This is, well, this could be good or bad depending on how this type of systems operate. And I'm not sure if, again, this is regulated, because one could say, well, there are certain advantages of this, right? You could convey all kinds of information because you don't have a limit of the label. So it is good from this perspective. You can give consumer plenty of information about your product, which you wouldn't normally be able to put on the labeling system. So that's good. On the other hand, are those systems safe? Is it hackable, right? Maybe you can have a particularly well-identified list of ingredients and everything is fine, and then suddenly somebody comes with an idea, okay, what if I substitute this list of ingredients by the list that shows that this is a better product than it is, right? So it's, let's say, not fake crab, it's real crab, right? Or it's not this, but it's that. And then if I'm caught by the authorities, I will say, well, somebody hacked the page, right? I had the QR code. How can I be responsible that somebody hacked the other side and misinformed the consumer? So it's a question of validation, right? So that's the danger, I think. But in general, I would say that, again, if you combine this with public key encryption technology and blockchain, you can have systems like that would be very safe and would allow for more information being transmitted to the public, which is ultimately good, because then manufacturers wouldn't be able to sell, we don't want another regulation that tells us that we have to mention every ingredient, even if the amount is, let's say, in parts per million, because if FDA asks us to do that, we would have to have a label which is 1 mile long, right? So now they have no excuse, because FDA may say, okay, put it on the website. But it has to be accessible. So from this perspective, I think that it's probably on the balance of advantages and disadvantages of this approach. I would be rather for it.
Last question, red microphone, unless there's somebody.
My name is Al Ehrlich. I am a member of this society. I don't think there are all levels of fraud. Some of them basically benign, like the fish you just talked about. Some of them could be quite serious. What is your process of reporting this information? To whom do you report it? Is it all treated with the same urgency, even though one could be terrible and one could be benign? And how does the information get out, and how long does it take? I mean, that whole process is
Yes. Important. So this process is unfortunately fraught with problems. And probably all of you at some point within the last several years were in fact witnesses to those problems. At least I was many times. When I go to a supermarket, I buy, let's say, peaches, and then a week later, no, 2 weeks later, sometimes, I go to the same supermarket, I buy peaches again, and then I take the receipt. And on the bottom of the receipt, instead of being this long, is this long, and there's this long printout saying that, oh, by the way, those peaches, which I bought 2 weeks ago, right, they were found to be contaminated, and I have to dispose of them as soon as possible. Well, I dispose of them by consuming them, obviously, a week earlier. So this is absolutely useless type of information to me. But that's what is happening now. And the reason for this, the reason why, for instance, food recalls take so much time and they are ultimately ineffective, is that everything is analyzed in a centralized system, right? Everything is sent to some central lab. Specifically with the food-borne contamination type, the standard is you always have to take it to the level to even start a recall where you can actually grow the pathogen on the Petri dish, right? Then you see that, okay, it's grown, it's identifiable, then you can start the recall. Before dealing with food fraud project, I mentioned, I think, during my talk that I worked for a few years on the side of identification of foodborne pathogens. And what we were trying to build was the system that speeds up the process of identification, right? Dramatically speeds it up. And we talked to some food manufacturers because of the contacts that we were given through USDA. And the biggest problem was that if you don't grow the darn pathogen on a petri dish, you cannot be 100% certain that it is really the cause, right? And there is always this tiny, tiny percentage of probability that you will cause false alarm. So every major food retailer and manufacturer, when they heard that our specificity is not absolutely perfect and then could be a tiny probability of a false alarm, then that's it. They are throwing their hands up and say, no, we don't want to implement the system. We don't want to know. Because by law, if we have a system, that's how it kind of operates with food industry, that if you have a process, you identify the process to FDA, USDA, and then you have follow it, right? You have quite a leeway on what is your process for identification of food safety issues. But if you declare that you have a process and this process takes the food product through the analysis, and if the process identifies possible contamination, then you are obligated to tell authorities about it. If you, let's say, run a food processing plant, or if you run Jack in the Box or McDonald's or whoever, you have to identify them. And then there is a procedure. You have to shut down certain things. You have to call a recall, right? So if there is a possibility that it's a false alarm, the cost could be in millions, right? So now you look at the cost. Okay, what is the probability that it's an adverse event, right, in terms of the actual foodborne pathogen? And if it is, how many people will be affected? And what are our legal costs? On the other hand, what are our legal costs if there is, let's say, a 100% chance that this is false alarm? And in all of those cases, they identified that the legal costs associated with the actual adverse event would be lower. Pardon?
If you find something, who do you call first?
Well, if, as I said, we typically study things which are not. It's not like we take stuff from a retailer, right, and we are looking for contamination with heavy metal, because obviously we could, wait 2 years before we find something. We typically use spiked samples, so we have a pure sample and then we contaminate it ourselves, right? So it's not this scenario in which we would have the obligation of telling anybody that we found something. But of course, if that happened, if, for instance, I was doing research on spices and I decided, okay, I'm going to Kroger to buy some spices just to see what's going on. And if I found in those spices, let's say, heavy metals, then to be honest with you, I haven't thought about it. Whom would I call? Probably I would call my colleagues at USDA who are administratively responsible for managing this grant, because I know them personally, right? I would call and say, what am I supposed to do? Because obviously there are also legal consequences of that, right? I definitely would not start by, going on Twitter and saying, oh, there's heavy metal in spices from a particular retailer. But this is a very specific case because we are not a lab that does monitoring, right? We are developing methods. So the probability of us stumbling upon some situation like that is very, very low. Now, labs which are mandated to do that, they have their own chain of commands, so to speak, and they report to the authorities according to this chain of commands, so they know what they do. But they received routine samples from the retailers, from the food manufacturers. So this is kind of a different story, right? We are outside of this chain because we are purely research lab. We are not a USDA lab that works on food quality in the context of the actual food that you can buy in the stores. Oh, well,
where does he shop?
So that okay. So let me put it this way: when I go to food science conferences, and sometimes we go with colleagues out for dinner, and they all think that I'm completely, utterly insane, because I go for dinner with the food science colleagues, and then I order, I start with a dozen oysters, and they all think that I'm suicidal, right? Because nobody who works in food safety does that. But, we should, it doesn't matter how long we live, it matters how intense we live, right? And the degree of happiness. So that's that's my approach.
All right, so I think we'll wrap it up there and thank our speaker, Bartek. Thank you very much.
Thank you.
Before you go, we do have 3 tokens of appreciation for you real quick. We have a framed copy of the announcement of your talk, a ribbon for you to wear wherever you go and tell people all about PSW, and a signed copy of Volume 1 of the Bulletin of the Philosophical Society of Washington. I think you'll find it interesting.
Thank you very much.
Thank you.
Thank you. Thank you for listening to me. Thank you for your invitation. It was really pleasure to address you all, and it's fantastic to be on the list with all of the other illustrious speakers that you were hosting. Thank you.
Before you all go, those of you who are still here, we do have some important announcements about the schedule.
Mm-hmm.
Please note that the schedule has changed. As you'll see here, we are not going to have a meeting on February 23rd. We have shifted the meeting to later in the year, and instead we're going to have a meeting on June 28th in the hopes of accommodating a speaker who said that they could only be here in the spring on June 28th. The next speaker is going to be Eric Klein. Some of you may know Eric from his previous engagement here or may have read his books. So Eric is an archaeologist. He will be here to do the 2491st meeting on March 8th. The speaker, he wrote many books on the Mediterranean Bronze Age collapse of civilization, and he's written a new book on what happened after the collapse of the Bronze Age civilizations. And he draws some lessons from studying the civilizations post-collapse that actually did quite well, and the ones that disappeared and are known only from the distant archaeological record. He previously spoke at the 2351st meeting on October 9th, 2015 on Sailing the Wine Dark Seas. And the video of that talk has been watched over 46,000 times. He's a great guy and he gives a great talk. The 2492nd meeting will be on March 22nd, and the 2493rd meeting will be on April 5th. And if we're lucky, Nima Arkani Hamad will get back to me and commit to speak on those dates on one of them with his graduate student, but he has not replied as yet. Now note that the dates for the next 2 are reversed from what I incorrectly told you in the past. The 2494th meeting will be on April 19th, but the speaker will be Richard Pyle. He is with the Bishop Museum in Hawaii. He'll be speaking on deep ocean corals. And he will probably tell us about the equipment that he developed to allow him to study the corals as a free diver at depths that are generally inaccessible to free divers, which is to say with scuba equipment that you can just swim around in for a long time. The 2495th meeting will be on May 3rd. The speaker will be David Spergel. David is a physicist, and currently he is president of the Simons Foundation. He was head of the Flatiron Institute. And I think he's going to speak on UAPs, unidentified aerial phenomena. But we'll see. The 2497th meeting will be on May 31st. The speaker will be Masaharu Subakura, and he'll be here to tell us about health effects of the Fukushima nuclear disaster, which involve not only the direct effects from the disaster itself, but also the effects of the various public health measures that were undertaken in the interests of protecting the population. The 2,498th meeting will be on June 14th. The speaker will be the former NASA Administrator Mike Griffin, who is currently co-CEO of Logic, spelled with a final Q. He recently testified for a different scaled-back approach to land the U.S. on the moon as soon as possible, which has been variously panned by many people in the space industry, including Elon Musk. They don't get along. And then, so we'll see. We will, of course, post commitments to the website as soon as they are firmed up. And so please check the website often to keep up with the schedule. Let's thank all of the people who worked tonight to arrange everything and to run the cameras and stuff. Thank you all. Please return your name badges to the plastic bins in the back, and I will now accept a motion to adjourn the meeting. A second? All in favor? And anyone opposed? Meeting is adjourned to the social hour.